is Life Cycle of Polysiphonia from Unit 3 Phycology for BSc Semester 1 Botany students. Polysiphonia is a red algae that belongs to the class Rhodophyceae. The order is Ceramiales, family Rhodomylaceae, and genus Polysiphonia. First, I will discuss some of the important characteristics of the class Rhodophyceae. Rhodophyceae are also known as red algae, and these are mostly marine, found in seawater. The color of the thallus or plant body is red due to the abundance of the pigment R-phycoerythrin. Some other pigments found in red algae are chlorophyll A, chlorophyll D, some carotenoids and phycobiliproteins, R-phycocyanin and R-phycoerythrin. The flagellated motile stages are absent. The vegetative cells, gametes and spores do not have flagella. Reserve food is Floridian starch, which resembles the branched glucose polymer amylopectin of true starch. Fluoridosides are glycerol, glycosides, mannitol, dulcitol, etc. are also found as reserve food. The cell wall is made up of cellulose and pectin. Calcareous red algae have calcium carbonate in the cell wall. Pit connections are the cytoplasmic connections which are present between the adjacent cells. Sexual reproduction is advanced oogamous type. Male gametangia are known as spermatangia, and the non-motile male gamete is known as spermatium. Female gametangia is known as carpogonium. The carpogonium has a swollen base containing the female gamete or egg. The long neck of the carpogonium is known as trichogyne. Genus Polysiphonia is a marine alga. It is found in seawater. It is lithophytic or epiphytic. That means it is found on other plants, aquatic plants, or on the rocks. Polysiphonia fastigiata is a semi-parasite. It is found on Ascophyllum nodosum. 16 Indian species are reported. Common species are Polysiphonia variegata, Polysiphonia urceolata, and Polysiphonia platycarpa. If we see the thallus structure of Polysiphonia, it looks feathery in appearance. The color is brownish red or purplish. The thallus is heterotrichous. That means it has an erect system of branching and a prostrate system of branching. Both erect and prostrate systems are polysiphonous. They have a central siphon and 4 to 24 pericentral siphon. The prostrate system is attached to the substratum with the help of unicellular rhizoids. The end of the rhizoids have finger-like projections or attaching disc with the help of which it attaches to the substratum. The erect system has two types of branches. Branches of unlimited growth are known as long branches, and branches of limited growth known as trichoblast are dichotomously branched and uniseriate. Uniseriate means these have single row of cells. Male and female gametangia are also present in these trichoblasts. The life cycle of Polysiphonia is peculiar. The alternation of generation or life cycle is known as triphasic diplobiontic. Triphasic means 3 phases are found in the life cycle of Polysiphonia. These are gametophyte, carposporophyte, and tetrasporophyte. Gametophyte is haploid, whereas carposporophyte and tetrasporophyte are diploid stages. The male and female gametophyte are different thallus. In the male gametophyte, male gametangia or spermatangia are present. In the spermatangia, non-motile male gamete known as spermatia are formed, which are released, and with the help of water current, they reach the female gametangium. The female gametangium is known as carpogonia, present on the female gametophyte. It has a swollen base containing female gamete or egg and a long neck known as trichogyne. The spermatium or male gamete attaches to the neck or trichogyne. The male nucleus enters the carpogonium and fuses with the egg nucleus, forming a zygote. Post-fertilization changes are very elaborate in red algae, and after post-fertilization changes, the second phase, carposporophyte, is formed. Carposporophyte develops on the female gametophyte after fertilization. It has a fruiting body known as cystocarp, and the wall or pericarp is the fruit wall of cystocarp. Cystocarp has an opening which is known as ostiole, and inside the cystocarp, carposporangia are present. In each carposporangia, a single diploid carpospore is formed. This carpospore liberates through ostiole or the opening, and it germinates to form the third phase, the tetrasporophytic stage. The tetrasporophyte is morphologically similar to the gametophytic thallus, but it is diploid. Gametophytic thallus are haploid. Tetrasporophyte have tetrasporangium. In the tetrasporangium, after meiosis or reduction division, 4 tetraspores are formed. These tetraspores may be arranged in tetrahedral manner or cruciate manner. The tetraspores are haploid. Out of the 4 tetraspores, 2 form the male gametophyte and 2 form the female gametophyte. So if we see the diagrammatic representation of the life cycle of Polysiphonia, it is triphasic and diplobiontic. 3 phases are gametophyte, which is haploid, carposporophyte, diploid, and tetrasporophyte is also diploid. Male gametophyte has male gametangia or spermatangia, in which male gamete spermatium is formed, which is haploid. In the female gametophyte, female gametangia known as carpogonia is present. In the carpogonium, female gamete or haploid egg is present. After the fertilization of male nucleus with the egg nucleus, a zygote is formed. The diploid zygote germinates on the female gametophyte, forming a second phase, carposporophyte. Carposporophyte is a diploid stage. In the carposporophyte, cystocarp has carposporangia. Carposporangia form diploid carpospore. Carpospore liberates through the ostiole and forms the third phase, tetrasporophyte. The tetrasporophytic thallus is similar to gametophytic thallus, but the tetrasporophyte is diploid. In the tetrasporophyte, tetrasporangia are present. In the tetrasporangium, tetraspores are formed after meiosis or reduction division. 2 tetraspores form the male gametophyte and 2 form the female gametophyte. So, the life cycle of Polysiphonia is triphasic diplobiontic. I hope this lecture is helpful to you. Thank you so much.